So hi everyone. Yeah, I'm Stephen. I'm here from Refine. I'm a product manager, and I want to talk to you today, continuing on the theme of having very small amounts of sample to work with, and how we can help you de-risk early and accelerate analytics and shorten timelines, removing method development steps using Refine's technology, mass photometry. So, as has been mentioned, aggregation is an important CQA, and techniques for this have been well established with mAbs, but we know with some of the newer modalities this can be challenging. This could be due to column clogging, due to the presence of large aggregates, skewed UV absorbances when doing SEC-UV because you have a different molecule in there which isn't protein or some RNA or DNA. Interactions between the drug and the column, and demanding samples. So our solution to this is using mass photometry, which is a technique which measures single molecules in solution, so no need to do column optimization, crucially with nanograms of sample, so we're generally measuring at around 10 millimolar in minutes. And I just want to stress again, we're measuring mass here, not size, so it is very much a mass measurement. The way this works is we're a scattering microscope-based technique, and we shine light upwards through our objective, and you pipette 10 to 20 microliters of sample onto this glass cover slip, and your protein molecules bind to that glass, and when they bind, they scatter some of that light. And so what you can see here is these molecules landing. They're scattering light, and then we record the interference between this scattered light and the reflected light, and we get these beautiful airy disk patterns here, and each one of those is one protein molecule. And then the contrast of this molecule against the background, so how dark it is against the background, very linearly correlates with the mass of that molecule. So using a simple calibration of a molecule of known mass, we get this beautiful intuitive output, which gives you counts on one axis and mass on the x-axis. So it's very, very easy to understand and interpret the data. So to give you an idea of how this looks, you open up the cover, you pipette on some sample, give it a little mix, and those molecules are starting to bind to the glass. And in a second, the camera will pan up, and you'll be able to see dots appearing at the top of the screen. And each one of those dots is one protein molecule landing on the surface, and by looking at the darkness of that molecule, basically, we quantify its mass. So it really is very powerful single molecule technology, which gives us this advantage of being able to work at nanomolar concentrations. And then, yeah, this is the output. So for an antibody sample, if you're interested in aggregation, you can very clearly see baseline resolved monomer, dimer, and a small amount of trimer there, populations in your sample. So how can we use this, these very, very small sample volumes to de-risk early? So what we have here is an output from size exclusion chromatography, and this is the UV readout from that, and we can see we've got the main product population here and two high molecular weight species here. So what we did is we took those fractions, and those have been, the people who wrote this article took those fractions and looked at them on the mass photometer. And we've got the main product here, which is a nicely monomeric species. And then in the high molecular weight 2 species, we've got this the drug product in a smaller amount, but then we've got this large dimer population here. And then for the high molecular weight 1 fraction, in the light blue, so this is measurement in PBS, we've got a tetravalent species with some other products. Now, one of the other nice things about mass photometry is that it's very buffer agnostic. So this gives you the power to try out different stress conditions very easily without having to do any kind of complex method development. So they've then remeasured these 3 fractions in acetic acid as a stress condition, and they've been able then to assess the amount of non-covalent species across these 3 different fractions, which could be important in terms of immunogenic response. Again, in this example here, the collaborator was looking to de-risk their sample early, so wanted to test a range of different antibodies under 3 different stressor conditions and in triplicate. And so with candidate 1, we're very easily able to see, using the mass photometry data, that this one fragments, and candidate 2, under certain stress conditions, has a propensity to aggregate and form a dimer and a small amount of trimer. The key thing, though, is that this is able to be done with only 7.5 nanograms of sample per measurement, so far less than with size exclusion chromatography, and also 3 times faster. So these measurements take 1 minute. We record a 1-minute movie, and then you have your mass histogram ready for you to analyze. In this example, this is data from Rick Biologics, and they wanted to, again, look at some different stress conditions, they're looking at different clones in this case, for biosimilar comparability. So the original molecule down here, and then some different clones. And they ran this on both mass photometry and on SEC-UV. And there's quite nice agreement, but in the results showing that clone 8 and clone 9 are the most similar in terms of their propensity to aggregate, with a much lower dimer populations in both the MP data and in the SEC-UV data. But when they did this, they reported that MP was 24 times faster than the SEC measurements that they were doing and consumed 300 times less sample, possibly allowing them to do this study much earlier when they have a lot less sample to work with. And where we compare across different column-free technologies, which are important to do as an orthogonal method to column-based technologies, we can see that MP has major advantages against both AUC and DLS. So we're delivering, in the MP data, you can very easily see baseline resolved fractions for the dimer, trimer, and tetramer peaks, which you can make out in the AUC, but is a lot less clear. And the DLS, you're really just seeing that there is some aggregation there with very few details in terms of what's actually in your aggregated populations. But the key thing is the amount of sample you're using to do this with, you're doing this with. So 30 nanograms for mass photometry versus 250 micrograms and 65 micrograms for DLS and AUC. And crucially for AUC, the time taken is huge. So we're delivering kind of AUC or better results with the simplicity of a method like DLS, where it's kind of pretty much you put your sample on, you hit record, and you have your data. We also, as well as being pretty buffer agnostic, we're also quite sample agnostic, and so can measure a large number of different types of antibodies and lots of other different types of proteins as well, without the need for extensive method development. So we have results from monoclonal antibodies, different sizes of bispecifics, and this example of ADCs. So you can assess all kinds of different modalities without having to optimize your column choice or your buffer choice, for example. Just as a slight side note, this ADC experiment is quite nice. So here they have just the antibody by itself, and then with the drug conjugated, we can actually calculate an average DAR here using the mass shift of that peak, again, one of the advantages of delivering high-resolution mass data. Another use of mass photometry in the antibody space is in cell line development. And because we are working with such low concentrations, we're able to assess the types of concentrations you would typically see in cell line supernatant. So you can, if you have problems, say, in your cell line development, you can put the supernatant on the mass photometer, and you're able to see whether your cell line is expressing, or whether, in the case of the control here, there is no antibody peak. And you would also be able to see a dimer population again if that were to be present. So all of this data was recorded on the 2MP, which is our flagship mass photometer that we make at Refine. So this will do kind of all different types of biomolecules, antibodies, other types of protein, but also nucleic acids as well, if you are able to polylysine your glass surface. We also have an automated version of this product, which is fully upgradable from the manual system. That will do 24 measurements in an automated fashion from a 96-well plate. We also kind of offer the full solution, so we have comprehensive service agreements. We have a range of consumables, so we provide the glass surface and cassettes you need to do the measurement, but also our own range of calibrants, so you don't need to find your own calibrant of known mass. We can provide one for you and provide quality metrics to go with that, so you know your calibration's good. We also have a suite of software products. So we have our acquisition software, we have a general purpose analysis software, and then most recently we've brought out a specialist software for antibody aggregation, which is part of our Streamline MP platform. So to go into that a little bit more detail. This is the antibody aggregation module, and basically this takes all of the effort out of doing the stats on your repeats. It delivers you robust method development, so you can just apply the same analysis to lots and lots of different measurements very, very easily. So it means you get robust data, and you're not, your results aren't varying depending on who's analyzed them in what way. So this is a very powerful tool for people who want to do a lot of measurements. So in summary, why choose mass photometry for your antibody aggregation assessment? You can de-risk early due to the very, very small amount of sample that we need and the incredible fast time, measurement time that we have, allowing you to get instant results. We're a column-free technique, which means we can fit very nicely orthogonally to column-based techniques. And we have intuitive data analysis, and I would also add it's very easy to use. Some people say a child could do it. We say our financial director can do it. Yeah, it's very simple. If you can use a pipette, you're good. And it's also a benchtop instrument. We've got one at the stand. It's about this big. So it kind of goes into any lab very nicely. The automated system is also benchtop and not crazy big either. And, and crucially, low cost per measurement, so we're well under $5 per measurement. We have a range of different instruments which we sell now. So the 2MP we've talked about today. I also want to touch quickly on the SAMX, because I know this is a mass photometer purely designed for AAV empty full characterization, and I know there's been some AAV talks today, so I wanted to touch on that a little bit. We also have a new product called the Carita MP. This works a little bit differently, and it uses a method we call macro mass photometry. And that's for large viral vectors and VLPs. We're happy to talk to you about that at the booth if you want more information on that. But to give you a little bit more information about the SAMX. So this broadly comes with all the advantages I've talked to you about in the context of antibodies, but the beauty of the SAMX is we're really using the mass detection capabilities, where the AAV, of course, isn't changing in size when you add the payload to it. So It's an empty AAV is the same size as a full AAV, but the mass has changed, and that's what we're seeing. But crucially, unlike many techniques, we also deliver enough resolution to for you to assess empty, partial, and full populations. So you may be having to go to techniques like CDMS or AUC to get that kind of information. Otherwise, both of which use a heck of a lot more sample and crucially require much more skilled operation. This product specifically also comes with 21 CFR Part 11 compliance, so we can go into GMP manufacturing and QC environments. A little bit about Refine. We were started 7 years ago as a spin out from Oxford University. We now have 500 instruments worldwide, spread over 30 countries. We have 1,500 publications now involving mass photometry, and we're now in 18 of the top 20 biopharm companies worldwide. I would say come and find us at booth 34, but you don't really need to find us because we're literally the one as you walk into the stand. So you'll kind of walk into us if you're not looking where you're going. So yeah, please come and have a chat. We'd love to speak to you about how we can maybe help you address some of your needs. And yeah, if you're just interested to talk about the tech generally, we'd love to have that conversation. But yeah, happy to take any questions.
I'm curious, like you say you measure the mass in solution. What about like if the aggregates already form particulate? Are you able to measure the size of the particulate, or I have to dissolve it in certain way?
Are you talking about like larger aggregates?
Yeah, yes, like particles.
So yeah, that you'd have to use a different technology for that. So generally, where people are using mass photometry, it's for kind of looking up to tetramer, pentamer,
okay,
rather than kind of very large aggregates, which you might look at.
And is that in native solution, like say if there's the map, say is conjugated with some other chemicals, non-covalently, can you measure the mass difference, like without interrupt the interaction?
Yes, but it would depend on the mass difference that there is. So for an antibody, you would need a mass difference of probably around 30 kilodaltons to be able to resolve that on the mass photometer. But if I just go back to this example here with the ADC, where the drug's conjugated on it, that's kind of that's within that resolution limit, but because we're doing kind of like an average DAR here, you are able to see a mass shift between the product with no drug and the product with drug. So there we can go a little bit smaller than that 30 kilodalton.
Okay, thank you.
Thanks.
Yes, I have a question about dilution. Sometimes you have to dilute the sample, and one of the points I see is can you really mimic then also the formulation conditions, because that sometimes is not always easy.
So yeah, a couple of different things here, I guess. So one is, I guess, mass photometry offers you something unique in that you can measure aggregation at those lower concentrations, which you might not be able to with many of the other techniques. However, if you do want to measure kind of in the micromolar, we do have a, sorry, I have to flick back through the slides again now. So we do have an add-on product which you can use if you want to assess in the micromolar. And how this works is, rather than just having that well which you pipette into, you have this microfluidic add-on, and you put a chip on top of the mass photometer, and we measure in flow, and there's a dilution mechanism on that microfluidic chip. So you're measuring the sample within 37 milliseconds of it being diluted every time. So the complex or your aggregates don't have time to disassociate. It's a little bit more time consuming than a standard mass photometry measurement, but no worse than size exclusion chromatography, for example. So that is there if you want to work in those micromolar concentrations as well.
In the beginning, you mentioned something about the principle of the instrument. How different is it from DLS? Because I remember there's some resemblance, but with DLS you can measure the size but not the mass.
Yeah. So it's very different to DLS from in many respects. I used to work on DLS when I first started in this industry. So if I just go to here. So with DLS, you're measuring, you're looking at the sample in bulk, and you've got all the molecules kind of floating around in the Brownian motion, and you're looking at how the laser intensity, which you're shining through there, how the intensity of that fluctuates based on how fast those molecules are diffusing, and they'll diffuse faster if they're smaller, slower if they're bigger. The only similarity between DLS and mass photometry is the fact we're using the fact that those molecules scatter light. What we're doing here is we're using the intensity of that scatter from an individual molecule, whereas with DLS, they're not using the intensity of that scatter, they're using the fluctuations caused in the bulk. So this is single molecule, DLS is in the bulk solution, and that allows us to get much higher resolution. And then the fact we're measuring the intensity of the scatter and not just how the light is interacting with all of those molecules together, it's very substantially different to DLS. Yeah.
And from the graph, can you actually deduce the molecule weight distribution? Is
Yes.
it really quantitative?
Yes, yeah.
Okay.
So, our mass repeatability is 2%, and our mass accuracy is 5%.
I always wonder, let's say, what's the benchmark if you look at such a graph? What can you compare it with to know this is the right outcome?
So I guess the golden standard, if you want mass measurement, would be native mass spec. We've some really nice papers with people like Albert Heck at Utrecht, where they've done correlative mass photometry with native mass spec, because there are some areas where basically mass photometry gives you a better overall picture than native mass spec, but obviously for identifying those really kind of like absolute masses, native mass spec is much better, and we get really nice alignment with the data, and the two can be used nicely together.
Okay, thank you.
Hi, Stephen.
Hi.
We have been having really a lot of fun with the refinement to MP. So I was quite surprised to see the difference that you could already capture for the antibody drug conjugate compared to the naked antibody. And I mean, that's because there's just one fixed DAR ratio in that sample, right? I mean, if there's a mix of different DAR ratios, would you still be able to capture it, or would it be just one huge?
So yeah, I think this just depends on a number of different factors. The molecule you're attaching and what the refractive index of that molecule is, because that will affect how heavy it appears in the mass photometry. What your average DAR is, so if you've got a large spread but with quite a lot of drug, then it will shift out more. And then, yeah, the spread, so it is very much an average. So, yeah, I would expect that what would happen is that peak would get wider if you've got a lot of variation, and will be narrow if you have less variation. But yeah, it is very much an average, and whether it's useful in that context very much depends on your situation. But this was a kind of nice bit of data which was flagged to us by one of our customers, so it's nice to show it off. Thanks.
Any more questions? All right, thank you again
Great.
for your
Thanks a lot.
presentation. And with that, we have about a 30-minute break. We will be back here at 4:20.